Hello, welcome back. Today we are going to discuss about the transduction process. So transduction is the transfer of the genetic material, that is the DNA, from the donor host cell to the recipient host cell by the help of a bacteriophage. So there are two types of transduction. First one is the generalized transduction and second one is the specialized transduction. So today we are going to discuss about the generalized transduction. So as you can see in this picture, this is the host bacterial cell, that is the donor cell, and this is the bacterial chromosome, and this is the bacteriophage. So the first step is the bacteriophage is attached to the bacterial cell, okay, and the next step is the penetration of the DNA will take place. Then because of the phase enzymes, it degrade the host DNA, the degradation of host DNA into smaller fragments. As you can see, there are smaller fragments of the DNA. So after that the replication takes place, and as you can see in this picture, generally the capsid, that is the phase code, it generally incorporated in by the phage genome, but in this case by mistakenly the bacterial genome incorporate into the phage code. So the phage with donor DNA, that means the phage with the bacterial DNA. Donor means the bacterial cell. So in this case the phage contains the donor DNA, that means the bacterial DNA, and that is called the transducing phase. What is the meaning of transducing phase? Transducing phase means a phase having the bacterial DNA. Okay. So in this case, during the maturation stage, instead of the phage DNA, there is incorporation of the bacterial DNA into the phage code, that is the capsid. Okay. So next step is this transducing phase then enters into the recipient host cell. So in the same manner, there is the injection of first the donor DNA. So the donor DNA incorporated into the second host, that is the recipient cell. Okay. So in the next step, as I said, the phase having the bacterial DNA is called the transducing phase, and it inject into the another bacterium, that is the recipient cell, by transferring the bacterial DNA into the new cell. So in this new cell there is a chromosome. So this phase, they transport the bacterial cell into this chromosome. You can see the blue part, this is the donor DNA part. So there is the incorporation of the donor DNA is incorporated into the recipient cell, recipient DNA, and this is by the help of recombination. So the donor DNA incorporated into the recipient DNA by the help of recombination. So this is all about the generalized transduction. So let me summarize this once again. First, the bacteriophage, it intact into the cell, then the penetration occur, then because of the phase enzymes it degrades the host DNA into the fragments. Then instead of the phase DNA, the bacterial DNA incorporated into the capsid, that is the phase coat. Then that transducing phase, that is the bacterial DNA transducing phase means the phase having the bacterial DNA. So is then inject into the another host cell with the donor DNA. So it then the donor DNA means the donor cell DNA then incorporated into the new recipient cell DNA and form a recombinant DNA by the process of recombination. So this is all about the generalized transduction, how the transfer of DNA takes place first from the donor DNA to the recipient DNA by the help of phase. In this case there is the transducing phase, that is the phase having the bacterial DNA. So this is all about the generalized transduction. If you like this video, hit the like button, share with your friends, and do not forget to subscribe this channel. In the next class we'll discuss about the specialized transduction. Thank you.